So welcome to this interview session with Dr. Don Cleveland. Dr. Cleveland is chair and distinguished professor of the Department of Cell and Molecular Medicine in the University of California, San Diego. And I'm very honored to talk to him today about his research and about the developments in molecular medicine that he has spearheaded. And Dr. Cleveland looks back at a long-standing and very proficient career in science. He has made seminal discoveries in the mechanisms underlying cell division. And he has also greatly contributed to our understanding of neurodegenerative diseases. And I think he will talk a little bit today about those ways of targeting these diseases. And he has made really encouraging development, just published also this year. And for his past research, he has won many, many awards. I don't want to enumerate them, just mention the Breakthrough Prize in Science, which is basically the Oscars of science. So welcome, Dr. Cleveland.
Oh, it's a pleasure to be here. Thank you.
Thank you. So to start, can you give us a little background about how you got into your research and what your current research involves?
So as a graduate student, I started in physics and and then really got interested in biology and switched into biochemistry, having never taken a biology course. And as my graduate project, I ended up purifying a protein. It became a famous protein, tau, which misaccumulates with inside the cells damaged and inside the neurons damaged in Alzheimer's disease. I just became excited about working in biology where individuals could make real contributions. It didn't take a huge team to do an experiment. It took a team of 2 or 3 or 4 could do anything. And it was just that it started that way. And then fast forward 20 years, there was the discovery of the first gene whose mutation is causative of the most frequent adult motor neuron disease. It's called amyotrophic lateral sclerosis. And that mutation, the clinicians and the scientists then working on the disease all said the mutation probably caused disease by a loss of activity of the gene. And we just set out, we looked at the mutations, that didn't seem to be correct. And then we demonstrated it wasn't correct, that the mutations encoded a protein that had gained 1 or more toxic properties. And then we thought, you could be directly on disease mechanism if you just turned off the disease-causing gene. And we looked at other examples of the human neurodegenerative diseases, the big ones, Alzheimer's, Parkinson's, Huntingtons, as well as ALS. And in every case, the genetics told us that the genes that were known to be causative or contributory were broadly expressed in the nervous system, and that in almost every case, the mutant gene did something bad. So if you could just turn it off, you would be directly on disease target. So we set out to do that starting in 2003. And we developed a new class of drugs. I call them designer DNA drugs. They're little snippets of DNA that when you infuse into the nervous system, you can deliver them very broadly throughout the nervous system, and they have amazing efficacies. So, when you take a typical drug, an aspirin, you get 3 or 4 hours worth of relief. But we learned that for these drugs, once you inject them into the nervous system, you get not 3 or 4 hours, not 3 or 4 days, not 3 or 4 weeks, but 3 or 4 months worth of efficacy of target gene suppression. So that just, as you indicated in the introduction, I was very pleased to have that work recognized by the receipt of the Breakthrough Prize in Life Sciences. Today, we have 1 proven example in a childhood motor neuron disease, and we have 7 ongoing trials, 3 in ALS, 1 in Huntington's. one in Alzheimer's, oddly enough, to suppress tau, the gene product that accumulates inside the affected neurons in Alzheimer's disease. Tau, the protein I discovered a long time ago. And lastly, just started earlier this year, 2 trials, one for an inherited form of Parkinson's disease and one for sporadic Parkinson's disease.
So in other words, even though these diseases are very complex, if I understand this correctly, but one protein gone rogue is enough to drive the disease development. And if you target that, you can cure or at least ameliorate disease.
We certainly hope we can do that. And actually, one of the primary discoveries was by a group at NIH, a guy named Andy Singleton. He discovered that if you inherit, you're supposed to inherit 1 copy of each gene from your mother and 1 from your father. But if you, because of a genetic quirkiness, you actually inherit 3 copies of a completely normal gene. You will get early onset Alzheimer's disease. Early onset Parkinson's disease, and that gene is alpha-synuclein. And just a couple of years later, it was discovered that if you inherit 3 copies of a different, completely normal gene, the one encoding amyloid precursor protein, you will get early onset Alzheimer's. So too much of a good thing. So gene dosage really matters. And so the strategy we set out is now to be able to reset that dosage by turning it down. So it's a good idea. Will it work? Yeah, so we're still we have good evidence, but not completely conclusive evidence in the first example in inherited ALS. And we have high hopes for other targets.
But these diseases are really fatal, and you don't really have any treatment. So I think a new treatment, even if it's not completely curing the disease, is a wonderful news for many patients.
You're absolutely right. And when we started this, and if there are any students in the audience, when we started this, everybody said the use of these DNA antisense oligonucleotides, designer DNA drugs, everybody said it'll never work. You'll never be able to distribute them broadly. And you could read any pharmacology textbook. They all said these drugs are way too big. They're about 15 times the size of a typical drug, and they're very heavily negatively charged because the DNA backbone. And all the books said that it'll never work. And the thing about it is that we tried it anyway. And it turns out that the cells of the nervous system don't read the textbooks. Or in other words, that the textbooks were wrong. The textbooks are wrong many times. And so that would be the lesson I would give any of the young folks. It's that read the textbook, test whether the ideas there are really correct. And of course, the breakthroughs are going to come for discovering things that we didn't know and probably didn't think would work the way they work.
And actually, you bet on the right target, because many pharmaceutical companies bet on another target, and their trials failed quite spectacularly, no?
Yes, indeed. And indeed, what we're really betting that gene dosage really matters. And that therefore we know that we have what the pharmaceutical companies call target engagement, because we just look for the reduction in the target protein that our drug is trying to reduce the synthesis of. And we know, and actually in every trial, we know we've done that. We've lowered the level of the protein product. And so we hope that in the inherited examples of age-dependent disease, you live with the mutant protein for 50 years, 60 years before disease onset. And in mice, we demonstrated that we could suppress disease progression by turning down the disease-causing gene, or in some cases, we could get disease reversal, where we interpret that to mean that that if you just lower the damage that's occurring, the neurons that are still there have a chance to recover and to rehook up.
That's really amazing. And I saw you published a paper in a mouse model where Parkinson's was cured, and you had one dosage given, or?
Yes, that was published in June this year in Nature, and it's I hope, I think, and I hope that it's the tiniest tip of a very big iceberg, where what we've showed we can do is to change cell identity, to use indeed a conventional antisense oligonucleotide designer DNA drug infused into the nervous system to change an abundant glial cell type, called an astrocyte, convert it into a replacement neuron for ones that have been lost to degenerative disease. And we showed that we actually could do that in an example of chemically induced Parkinson's-like disease in mice, where we generated new neurons. We got disease reversal in those animals that appears to last the lifetime of the mouse. So I couldn't be more excited about the potential of developing this approach now to marry the use of these designer DNA drugs and the discovery that we can change cell identity as a way to generate replacement neurons. Not just in that. Well, it would be super fantastic if it really work in humans Parkinson's disease.
Yeah.
But I'm hopeful that it could be much more broadly applicable to generate replacement neurons.
But I mean, I know personally because one of my family members was actually affected by dementia with Lewy body. And for just watching disease progression, and in the beginning, one doesn't even know what's wrong. It's really a slow onset, and then suddenly it advances quite rapidly. And he died 4 years ago. So I think there is really a kind of a deep involvement once one has seen the fate of such patients.
And absolutely, and those Lewy bodies are probably made of normal alpha-synuclein that just misaccumulated for some reason, and so we would argue that for that instance of disease, and actually most instances of Parkinson's disease where alpha-synuclein deposits accumulate, that maybe we can really slow disease advance, and then in the longer term, maybe we can even make replacement neurons. And this sort of comes full circle for me. So you just described a personal example of your own. I watched my father died from Parkinson's disease. I saw every aspect of that disease, starting from when I was about 10. And then there was this transformative change in Parkinson's disease therapy in the late now for someone like you and your students, it's going to seem like ancient history, but it was in the late 1960s when L-DOPA, the therapy was developed, the idea of replacing the neurotransmitter that's lost by the neurons that degenerate in Parkinson's disease, and just to feed patients a huge amount of it and hope that some of it gets to the right place. But it really changed life for Parkinsonian patients. And the truth is, we've had important further steps in the 40 years since then, but none were as important as L-DOPA. And I'm hopeful that actually, yes, replacement neurons could, in another half a decade to decade, be a true replacement therapy.
So in principle, your career really spanned the entire spectrum. And even with identification of the protein tau, which was early in your career, the basic science to figure out what this protein does, and also what you later worked on cell division, what the machinery involved in this process is, what the proteins do. Then you moved on to disease processes, and now you're hopefully also curing some of these diseases. And maybe I would assume that your approach is probably applicable to many, many different diseases, ataxias maybe also, no?
almost all the ataxias, and there are efforts underway for spinal cerebellar ataxia 1, 2, and 3. So yes, absolutely, there are going to be multiple efforts, and because those diseases are caused, at least in large part, by gene mutations that cause one or more toxicities, turning the bad gene down just seems like the right thing, as long as you can get your drug to enough of the right cells.
Yeah. So that is really exciting. And I remember I'm teaching, or I taught a course, problems-based course on gene therapy. And just incidentally, my students talked about how to cure Huntington, and they also discussed your approach or some of your approaches that you developed. And I think it's also quite exciting if the students hopefully listen to this interview.
We did show in all of the mouse modeling done with Huntington's that we could stop further neuronal loss, and in 2 examples that we could get disease reversal, sustained disease reversal from a single dose infusions to lower the synthesis of mutant Huntington. So, that's now in a large phase 3, the final phase clinical trial, carried forth by Roche, big pharmaceutical company, and I'm hopeful. I don't play any direct role in that, but I'm one of the biggest cheerleaders.
Well, phase 3, that is really advanced. That is exciting. So you used antisense, right? And now with the new method CRISPR, are you thinking about approaches using that method as well?
I certainly think CRISPR approaches are going to be part of the arsenal that will come into practice in human therapy in the next decade. In the nervous system, it's a huge challenge because the CRISPR methods for genome modification, I do believe that is the third of the transformative events that have occurred during my lifetime in science. The first was the discovery of restriction enzymes. They just changed almost every aspect of medicine and most aspects of biology. The second was DNA sequencing. Fred Sanger and his team in Cambridge, England, they developed a sequencing approach that people could do in their laboratories all over the world, and it just completely changed the scope of things that can be done. And I think the discovery of CRISPR-Cas9 to be able to take restriction enzymes one step further, to be able to cut, you get to choose the site at which your DNA is cut. That's the final of these transformative technologies that has occurred during my lifetime in science, and I'm excited by it. But delivery, making it happen in the nervous system, I think that's a ways off. But on the exciting part, that's where if I were a young student, absolutely, someone is going to make it work, or actually probably multiple teams are going to make it work in different ways, and the different approaches will have advantages in some cases, and a different approach will have an advantage in another case. So it's not the case that there's one person who's going to make a great advance. There are multiple people who are going to make that work. And it's super interesting.
Yeah, we see really what science can achieve in a very short time with COVID-19. I mean, the amazing collaborations, even between kind of adversarial pharmaceutical companies.
Yes.
So if we could translate that to CRISPR approaches and other approaches, I think.
We developed we did develop on one of the problems with the CRISPR approaches is if you turn on a nuclease that the CRISPR and it has high specificity for the site you want to cleave, but it's like using your scissors with great care. But if your scissors if you have electric scissors and they're running all the time, that's a little dangerous. So we designed a strategy which we have proven to work well in cell culture, where we can introduce the CRISPR enzyme and then use a DNA, using an oligonucleotide delivered the way that we've been delivering these other drugs, and now activating the CRISPR. And then that will then turn off as soon as our oligonucleotide is catabolized. So I do think that we absolutely need strategies for transient CRISPR activation, and methods to be able to deliver them broadly. And for the students, we don't have either of those today. So there's plenty to do.
I think you answered the question that I will ask you later.